How don't we have the eyes on below the nose or one instead of two? How very everything is maintained in a species or in individuals? So the answer is the gene expressions. The role of gene expressions is very critical in the expressions of specific traits in individuals at populations level. Gene expression is the process by which the genetic code, that means the nucleotide sequence and the nucleotide sequence, combinations of nucleotide sequence is called the genes. So the synthesis and expressions of genes determine the functionals and the end functions of the traits in individuals or in the populations. So the major concern is about the how do we identify those compositions of nucleotide ATCG, right? So and how the occurrence of ATCG informations controlling the specific functions of the traits, that means the characters of the individuals at populations level. So this is the fragments of DNA contain the ATGC information, that is the called DNA, and gene is the chromosomal regions which determine the specific functions or attributes of the individuals. Here is the basic examples of different characters or traits we can easily visible or in a population, or sometimes we cannot visible in a population, that are called the biochemical traits or biophysical traits
Sir,
or
your slide is not moving. It's not changing.
now it is changing.
So it's in very first slide only.
So I will stop my videos so that it can easily move. This slide is visible. The second slide,
So it's only the first slide is visible.
introductory slide.
No, sir. Rest of the slides are on side.
Ma'am, this is the my first slide. Is it visible?
Yes, the first slide is visible.
No. No. No,
Yes,
it is
sir.
moving, ma'am. Is it
Yes, sir.
moving? Hello. One moment. Where? This one? This one? Okay. One moment. Is it visible? Full slide? Is it movable? Okay, okay, okay. Sorry. Um, Sorry for the interruption. Uh, So I'm talking about the configurations of the genome in individuals. So as we all know, the genome is the total genetic information contained in a cells, which is composed of the four nucleotides A, T, G, C, right? So the question is here, there are many genes in every cell, right? So how they maintain and differ in functions? How don't we have the eyes on below the nose or one instead of two? How everything is maintained in individuals at populations level or at individual level. So the answer is the regulations of genes. Gene expression is the criticals, very criticals in the expression of specific types of traits or characteristics in individuals at populations level. So gene expression is the process by which the genetic code, that means the nucleotide configuration A, T, and GC, right? So the configurations of nucleotides in a segment is called genes. So gene is the expression of gene, and the function of gene is decided the functional and functional characteristics of the individuals. So now, major concern in the diversity analysis and in genetic, molecular genetic is to identify and characterize the genetic variation that is called ATCG. So, how do we identify the factors or nucleotide codes which controls the specific function or characteristics in individuals? So this is the fragments of DNA, and this is the ATGC segments presents on the DNA is called gene. A gene is a chromosomal regions which decides the specific attributes of the individuals. So basic examples of the traits or characteristics in plants as well as animals. So sometimes we can easily observe the characters of the traits in individuals and sometimes not, which is characterized under the biochemical traits or the biophysical traits. So as we can see here, very easily the short height plants and long height plants, high carotene levels in carrots and low carotene level carrots, which can easily detect in the in the individuals. But the question is how the color differentiations occurred in the individuals, how the heights of the plants occurred or functional in the individuals. In case of animals, we can we easily observe the color, hair colors, browns and black, such as the brown hair color, black hair color. So many examples in human individuals, uh, which differentiates the individuals to individuals or population to population sometimes. In a previous study about the cancers characters or cancers attributes in the populations, populations with cancers and population without the cancers. So is it determined or is it controlled by the genetic factor or at populations level or the influence of the populations to regulate the characters in a populations? So how can we decide or how can we characterize that those characters of populations is controlled by the genetic factor or by the environment influence? So diversity challenges, there is a major challenges in diversity analysis is the environmental consequence, how the climate changes interplay between the prevalence of disease or the adaptive strategy of the individuals in ecosystems. And the second challenges is the ecosystem impact, how the social ecosystem that is called the soil health, microbiome, the diversity influence the diversity and diversity of the populations. And the nature of the disease that is called the biotic disease, how the disease deteriorates the quality of the plants or increase the metabolic disease rates in the populations. And the fourth very important challenges is the genome complexity. As we know, the diversity analysis, the understanding of the genetic configurations of the individual is very important. And the genome complexity, as the polyploidy levels, diploid, tetraploids, and hexaploids. So increase the ploidy levels is directly proportional to the genome complexity of the individuals. How the certain variables affect the populations during the diversity analysis, and can we integrate these types of study in our diversity analysis? So yes, the measures certain variables is the natural selections, mutations, genetic drift, gene flow, dispersal barrier, biotic and abiotic interactions, and environmental marginity. So here is the pipelines to identify the genetic variants, and what are the initial data requirements for the genetic analysis of the individuals. So first one is the phenotypic data, that is the trait data. Second one is the genotypic or marker data, and third one is the population structure analysis, and the most important one is the kinship data, that is called the pedigree analysis. These are the flowcharts. You can browse these flowcharts in the previous paper published by us. So now I'm coming about the NGS technology. What are the importance and how the NGS advancements occur during the last 5 decades? As we know, in 1953, the discovery of the structure of the DNA, and in 1977, the first genome sequence is drafted by the first generations of the engine sequencing technologies. In After that, the second generation technology came by the different companies marketed by the Applied Biosystems. And recently in 2004, NGS came, and when NGS came, a tremendous revolutions occurred in the sequence technology to analyze the diversity. Hello. Oh my God. So which slide is visible there? Can we please hints? Sorry.
हाँ
So which
ये
one slide
दिखा
is
तो
visible?
मतलब वो
Can
नहीं आ
please
रहा है.
hint me? Which one slide is visible
अच्छा
right now?
ठीक है दादा, ठीक है. ओके 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 ओके.
The challenges in different in diversity analysis.
Okay. Okay, ma'am. Thank you. So should I repeat the slides or continue from the next slide? Am I audible? Ma'am, am I audible?
Yes, sir, you are audible.
So should I continue the slides or can...
Yes, sir, please continue.
Okay. So I'm talking about the advancement in sequencing technology. Sequencing technology slide is visible. Advancement in sequencing technology slide is visible, ma'am.
Yes, yes, sir.
Okay, so I'm talking about the advancement in sequencing technologies. So as we know, the first draft, first in 1953, DNA was DNA double helix structure was established. After that, in 1977, first generation of sequencing technology came, and then second generation sequencing technologies, and in 2004, the next generation sequencing technology came, and after that, tremendous revolutions occurred in the sequencing technology. So what are the advantage of next generation sequencing technology? So we can employ our very minute amount of DNA samples in this approach, micro and nano amount of DNA samples. It is very low reagent cost and less time consuming. That means we can sequence parallelly experiments thousands of samples at a once and produce extensive amounts of genomic data. These are the technologies commercially available in next generation sequencing, that is called the Illumina, Roche, Helicos, Life, Pacific Bioscience, Anton. These are the companies which provides the sequencing technologies very easily. And in these, Illumina is a very popular in sequencing machines provider company, because these are they develops a variants of instruments for the sequencing technology, and all the instruments are specific to the maximum size output size of the genomes, and each instrument is have specific characteristics. So what can be done by the sequencing in diversity analysis? So we can easily done genome sequencing, either reference based or the de novo sequencing, targeted resequencing, whole genome resequencing, transcriptome sequencing, gene expression profiling, total RNA sequencing, whole transcriptome, that is coding with non-coding sequence. In this, recently, our epigenome sequence is integrated in NGS technology, that is called the epigenomes chromatin immunoprecipitation sequencing. That is a cheap seq. We can detect the methylation in a sequence, and we know the methylome sequencing provides the influence of the changes in ATGC due to the environments. So how the environments change the genome sequence at gene levels or the RNA levels. So for the detections of epialleles or epigenetic regulations, we can go with the chromatin immunoprecipitation sequencing. Now I will come on the how we can develop the marker, molecular marker, because the first step in diversity analysis is the developments of marker and the characteristics, characterizations of the markers. So in these days there are major molecular marker used in biodiversity analysis is the SNP marker, single nucleotide polymorphism. So there are two types of methods to retrieve the single nucleotide variations. First was the aligned to the reference sequence. When we have the reference genome, we can easily go with the reference sequence alignments. And if we have no, we haven't the reference sequence, so we can go with the de novo assembly, that is called is the transcriptome based or the de novo based. So there are two types of ascertainments procedure for SNP discovery, that is called the KPS approach, SSP approach. KPS approach is the Perl-based algorithm for the discovery, for the ascertainments of SNP in a specific fragments of the genome or genomes. But when we need in the whole genomes configurations or whole genome variations, so we should go with the whole genome or the high throughput SNP genotypings by the Illumina Golden Gates. Or there are so many approaches based on the your requirements or the types of complexity in the genomes, bead express assay, Infinium assay, and Illumina Golden Gate assay. So how can we discover the SNP? There are different steps or procedures in SNP discovery, such as the first is the very high quality DNA is very important in the SNP discovery. Second one is this is the protocols for the DNA digestions, ligations, and library preparations for the sequencings of SNP discovery. Now I come on my few previous research work for the diversity analysis and the characteristics of the different wheat genotypes. So we have collected so many more than 250 wheat genotypes all over the world to analyze the characteristics, starch-based characteristics of the wheat, and how the different ecosystems or environment influence those characters. So we have included all over. We have included the wheat lines adapted in all over the India, 6 agronomic wheat zones. And as we follow the procedures of the DNA extractions and Illumina high-seq library for SNP callings, these are all the protocols for SNP calling. These are the computational algorithms used for SNP calling or reduce the number of high quality SNPs. How can we extract the high quality SNP, which have the major influence on the traits or the characters of the individuals in populations level? So out of so among the 250 genotypes, 3,691 genetic variations were identified, which influences the diversity of the different wheat genotype adapted in different agronomic zones in India, and the characteristics of those wheat genotype is very specific towards the starch quality. These are the marker characteristics used in the study, and these are the density of the SNP marker available in all the 21st wheat genotype in the wheat. So on the analysis, we can conclude that the regions belong to the Punjab, Haryana, Chandigarh, Western UP, Rajasthan have a low trace value. That means the not much important for the high quality chapati or high quality starch, but the individuals or genotypes, sorry, this is a arrow put wrong. The this trait value is adapted for the MP region, that is called Gujarat, Chhattisgarh, Udaipur, Rajasthan. So those regions have cultivated their high quality of wheat genotype. These are the interactions among those SNP identified for the specific character of the starch. So as we know, the when we talk about the characteristics of the trait, 80% or more than 80% characteristics is influenced or governed by the polygenic inheritance. So that means the controlling by the multiple genes by regulating in multiple pathway. So there is the interactions pathway between the genes influences the characters of the wheats specific to the starch quality. In another study, as we know, in recent advancement, we can retrieve the genetic variants by transcriptome analysis through NGS approach. So we used 713 T germ plasm collected from the different regions of Himachal and Assam, and identify the some SSR primers specific to the transcription factor. And we analyzed the diversity analysis of among 713 genotypes. Those all the 713 genotypes are distributed in six group. These are the characteristics of those markers developed by the specific to the transcription factor. Now, what are the statistics and software used in genetic diversity? So there are so many statistical models used in genetic diversity analysis. So it depends on the population's number, what are the complexity, and what are the targets of our study. And these are the softwares, popular software used in genetic diversity. So thank you.